think maybe we should make a start. Welcome everyone, and thank you all for coming. It's great to see such a large turnout for the first CNI seminar. It's my pleasure to introduce Ediz Sohoglu. We've been trying to get him to give a talk for about 8 months now. He was scheduled in the last 3 terms, but for some reason or another, be it strikes or pandemics or whatever, it's always been postponed. So we finally get to hear from Ed, who fairly recently joined the Department of Psychology. So if you could try and keep your mics muted, and if you have any questions or just want to register that you have a question, write it in the chat, and there should be some time at the end for discussion. So without further ado, over to you, Ed.
Thank you very much, Chris. Yeah, this is, we were just talking about it. It's very strange doing this from my bedroom in Hove. We'll see how it goes. I mean, I'm always keen to have questions throughout, but I'm even more keen in this sort of context. So I just feel like so I don't feel like I'm talking to myself. So yeah, I mean, Chris introduced me already, but just to say that, yeah, I'm still quite new. I've joined as a lecturer in the School of Psychology, for those of you that don't know me. And today's talk then is going to be about some work that I was doing as a postdoc. So before Sussex, I was the MRC Cognition and Brain Sciences unit in Cambridge. And so all this work is from my time there. Okay, so yeah, I'm going to be talking about how prior expectations influence perception. And I've given it quite a general title, but actually the talk's about speech perception. But I'd like to think that it has implications for other domains as well. Okay, so just to give you an outline of what I'll be talking about, I'm going to be talking, firstly, I'm going to introduce you to this notion of uncertainty in speech perception. And then I'll be talking about how and when prior expectations combine with sensory input. I'll be talking about some different models. And then the data itself, the experiments themselves, are all about using neuroimaging to test these computing models. Okay, so it's a bit of a dry slide to start with, but I think it makes the point I want to make. So this issue of uncertainty in speech perception, this is one of the main problems that the speech perceptual system has to solve. So to illustrate this, just consider doing a really, really simple experiment. Okay, so the subject comes in and the subject listens to different syllables and is told to repeat back the syllables. And so we can record the subject's responses, and we can look at certain speech sounds. So for instance, the sound sh and s. Now, one of the main acoustic cues to distinguishing these two sounds is acoustic frequency. And so we can look over all our measurements and construct these histograms here. So we've got 2 distributions, one for each speech sound. And you can see here that acoustic frequency is plotted on the x-axis, and you can see that, well, first of all, there's variability. I mean, I guess that's not surprising in itself, but more importantly, the 2 distributions overlap. So this creates a problem. So if you were to hear a speech sound that had a frequency of 5,700 hertz, how are you supposed to know whether it's a sh sound or a s sound? Now, this is, of course, from a purely auditory perspective, if you didn't have access to lip movements and other cues. Okay, so that's uncertainty in speech perception. There's many ways, potentially, that this can be solved, but one of the important ways it can be solved is going to be illustrated, hopefully, on this next slide. So let's play some sounds. Hopefully you'll all hear this. So firstly, I'm going to play you a sound of the words green needle. So have a listen. Turn up your speakers, please, and make sure you're ready. So can anyone just, at least someone confirm to me that they heard that? Yeah, Chris is giving a thumbs up, that's good. Okay, so I've just played you green needle. Next, I'm going to play you brainstorm. Okay, just one more time. This is a green needle again. And this is brainstorm. So I can see Chris smiling, so I guess he's expecting the trick that I'm trying to play on you all. So I've played the same sound throughout. It's exactly the same sound. I've only played one sound, but I've given you the expectation of green needle the first time I played it. And so hopefully you heard it as green needle. The second time I played the sounds, I gave you the expectation of brainstorm. So yes, I mean, the clue was in the title of this talk. So one of the main ways that the speech perception deals with uncertainty is by using prior knowledge or expectations. And as an aside, this all came about from this YouTube video here, apparently. So it comes from this toy. So this YouTube video is reviewing this toy. And apparently you press this button and it makes that kind of spaceship sound, followed by the word brainstorm. Someone on the internet has realized somehow that it can also be interpreted as green needle. And this made its way to social media, Twitter, TikTok, etc. And it caused a lot of excitement. I think you may also recall this Yanni Laurel phenomenon as well. It happened around 2 years ago, and it was a similar thing. So there was this ambiguous recording of the word laurel. Some people heard it as laurel, others as Yanni. So these are so-called perceptual illusions, but they, it's very handy for this talk to demonstrate the power of prior expectations on perception. Okay, so let's get to the science now. So this question, how do prior expectations combine with speech input? Well, the first place we can look is statistics, because Bayes' theorem and Bayesian inference tells us the optimal way to combine prior beliefs with the incoming data. Okay, so according to Bayes' theorem, you start off with some prior belief in a hypothesis. And can you see I'm going to be using my mouse a lot. I don't know if you can see my mouse. Yeah. Okay, great. So you start off with some prior belief, and it's all expressed as probability. So you have some prior probability for your hypothesis. Then you get some evidence in, some new data. And Bayes' theorem tells you how to revise your prior based on this new evidence to construct an updated prior, which is called a posterior. So the posterior then is the final thing you're left with. This is the final probability you're going to have in your hypothesis after you've taken into account both your prior beliefs and the new data. Okay, so many people think that this is what the brain does, or maybe an approximation to it. But the thing is that there's multiple ways to achieve Bayesian inference in the brain. So let's consider the first possibility. So the first possibility is this sharpened signals account. So according to this account, then, what neurons are doing is Bayesian inference relatively directly. So they're doing this this multiplicative operation. So the neurons are encoding the heard speech, so that's what the input is. And then there's also predicted activity patterns. So this might be expressed as pre-stimulus firing patterns. And so those two are multiplied together, and what you get out of that is a sharpened representation. So a sharpened representation of the inputs. And because it looks very much like Bayesian inference, you can interpret this sharpened representation as a posterior. Okay, so that's one way, but it's not the only way. So a second possibility is that what the brain is doing is computing prediction errors to compute to do Bayesian inference. And so instead of doing the multiplication, what you're doing is a subtraction. So you subtract predicted speech from heard speech to calculate prediction errors. Now, importantly, these prediction errors can be used to correct or update your predictions. And what you're left with is something that looks very much like a posterior. So in both these accounts, then, you get to the same place, right? You get to posterior probabilities, but the way it gets there is different for each of these models. And I think, an obvious question to ask is, with the prediction error accounts, it seems like a bit of an indirect way to do things. So why go to the trouble of computing prediction errors? And so this slide demonstrates this. So if you look at an image, and if you think in terms, purely signal processing terms, what's the most efficient way you can transmit this image? well, you can transmit the image as it is, and if you plot that, you're looking at over pixels, what the intensity is of the pixels. That's what's shown in the blue line here. Another thing you can do is to compute prediction errors. So here, what you're doing for the prediction errors is at each pixel, you're removing the mean from surrounding pixels, right? So what this is going to do is going to highlight bits of the image where there's edges, where there's a sharp discontinuity in the image. And so this is a form of prediction error computation. And if you now plot the intensity of those pixels when you're computing prediction errors, what's shown is in the red line, right? And as you can see, the dynamic range is much smaller when you're computing prediction errors. And so to transmit this signal, you're going to need a lot less energy to transmit it. So in other words, it's very efficient to compute prediction errors. And perhaps the brain is doing exactly this. So you've got a brain region, and instead of communicating with the next brain region by transmitting its representation directly, what it does is to compute prediction errors and only transmit the prediction errors. Okay, so that's the kind of the first issue. What about when prior expectations combine with speech input? Okay, so like with any perceptual system, speech perception is hierarchically organized. So you've got, at lower level stages, sensory stages of processing, and then as you go up the system, you have more abstract representations. In the case of speech, it's things like phonemes and words. But models disagree about whether processing is strictly bottom-up or whether there's top-down connections that are involved. So if you take an example like the merge model by Dennis Norris and colleagues, so I believe Alan told me this, Alan Garnham, that he did his PhD at the same time in the same lab as Dennis, so it's a bit of a Sussex trivia there for you. Dennis works at the CBU. He does make some important points, and with this merge model, this model can explain expectation effects without invoking any top-down processing. So according to this model, then, if you have a prior expectation of a word like brainstorm, that information enters the higher level, so the word level, but it doesn't modulate lower level phoneme processing, for instance. What happens is that all information gets integrated at a late decision stage without changing earlier level sensory processing. Okay, so merge can explain expectation effects without invoking any top-down processing. Alternatively, models like trace and predictive coding. Predictive coding is a model that has prediction errors in it. So these models have top-down connections. And so in principle, information at higher levels can modulate in a top-down fashion very early levels of sensory processing. Okay, so that's the second issue then. This existing models disagree about when prior expectations can combine with speech input. Okay, so just to wrap up then, what I've just said, all accounts make the same experimental predictions about the outcome of perception, that is listeners' behavior. They're doing Bayesian inference after all, but they do differ on the neural computations that lead to the perceptual outcomes. And so, yeah, in the experiments I'm going to be talking about then, is going to try and use neuroimaging to adjudicate between these theories. Okay, so in the first experiment, and I'm going to be spending most of the talk talking about this first experiment, we use not clear speech, but we use a very degraded form of speech called vocoded speech, and we present this to our listeners. So what is vocoded speech? Well, it looks like this. So in terms of spectrograms, so I don't know if you're familiar with this, spectrograms is one of the main ways to visualize sound. So we can look at spectral content on the y-axis here and how it's changing over time. So that's what a spectrogram is. And as you can see on the top panel, you have a case where vocoded speech only has 8 different spectral channels in it. So this is one of the nice properties of vocoded speech. You can parametrically manipulate how much sensory detail there is in the speech signal. So in the top case, where you have quite a lot of channels, 8 channels, you can see there's quite a bit of spectral structure, just as there is in clear speech. But if you reduce the number of spectral channels, so 4 channels or 2 channels, you can see that you're blurring the spectral structure. And so speech gets more and more unintelligible. Another motivation for using vocoded speech is that it may have implications for cochlear implant users. So this is a child who has a cochlear implant. and it restores hearing to an otherwise deaf individual. So the way it works is it uses a microphone that sits behind the ear, converts sound into electrical impulses that are sent to an electrode array inserted in the cochlea of the inner ear. And because this electrode array is making contact with the auditory nerve, this is how a cochlear implant restores the sense of sound. But importantly, there's a limitation to how many different parts of how many discrete parts of the basilar membrane the cochlear implant can stimulate. So just in the same way that in vocoded speech, you have these discrete, limited spectral channels. So that's the reason why vocoded speech is thought to be a simulation of what cochlear implant users hear when they hear speech. And just another thing to say about cochlear implants is, they are pretty remarkable, considering that if you look to vision, there are retinal implants, but they're still, they're not offered through the NHS like cochlear implants are. And that's because, they haven't really achieved the same sort of success. And, to date, there's more than 300,000 individuals who have cochlear implants. So they're really the most successful sensory prosthesis. And hopefully the research here has implications then for these individuals. Okay, so let's have a listen of what vocoded speech sounds like. So have a listen to this.
It was a sunny day and the children were going to the park.
So play one more time.
It was a sunny day and the children were going to the park.
So hopefully you'll hear that. Sounds a bit funny, sounds like a harsh whisper. And now I'm going to manipulate your prior expectations. So have a read of this sentence. It was a sunny day and the children were going to the park. Now, if I play it again, the same sound, it should sound a little different.
It was a sunny day and the children were going to the park.
So hopefully that sounds clearer with prior expectations. And this is exactly the sort of thing that we do in our experiments. So in the experiments that I'm going to be talking about, we present these single words. In this example, it's the word clay. And we present it with different numbers of spectral channels in the vocoder. So in this case, 3, 6, or 12 channels. And we manipulate prior expectations by presenting a written prime before the spoken word that can match with the speech or it can mismatch. And the task is to rate using this kind of rating scale from 1 to 4. How clear is the speech? So it's a subjective measure. And yeah, I put this here so I don't forget to say this because I think it's important that in our experiments, the spoken words are trial unique. So if we present a spoken word, the listener will never hear it again. Okay, so what about behavior? First of all, I mean, it's nothing really surprising here. So if you look at rated speech clarity, if you increase the amount of sensory detail, ratings go up. Makes sense. But just to confirm as well that if you if you give listeners matching prior expectations, that's shown in the green bars, clarity ratings also go up compared with when prior expectations are mismatching. Okay, so I've told you a little bit about the paradigm. Now I can talk to you in more detail about how we can tease apart these models here with this paradigm. Okay, so this study, I should say, is building on previous work. So by this study here by Blank and Davis in 2016. And so one of the nice things that this study did was to do some computational simulations of this paradigm, right? So it implemented the sharpened signal accounts, and it also implemented the prediction error accounts. And then what they did was to look at the representations of speech in the model and see, and they tested how things differed as a function of sensory detail and also as a function of prior expectations. And this is what you get. So for the sharpened signals model, you get these main effects. So representations are enhanced when you increase sensory detail and also when you provide matching prior knowledge. I think that's pretty intuitive. But for prediction errors, you get this funny interaction profile. And so I'm going to try and unpack this then on the next slide. So let's have a look. So let's consider the sharpened signals model first. So here on the left is what I've shown you in the previous slide. This is just summarizing the outcomes then of this model in terms of sharpened representations. And here then we're going to be looking at the different components of this equation, right? So let's look at the input, so the heard speech. For the mismatching condition, and so if you look at the inputs, this is what you see. So I should say, so in this depiction here, it's hard to visualize sound. So what I'm doing instead is visualizing things as written text. So speech is represented by this visual text here. And as you can see, as you increase the amount of sensory detail in the input, you go from a very blurry clay to a less blurry clay. All right, so that's the input. So that's this bit of the equation here. And then in this condition here, we have mismatching prior expectations. And so here we have the wrong word, so it's the word fast. And so if you multiply this with the inputs, this is what you get. So this is the sharpened representation. And as you can see, as you increase the amount of sensory detail, you have a bit more of the clay. You get what looks like a blend between clay and fast. And so this is why the clay representation gets enhanced as you go from low to high sensory detail in the mismatching condition. Okay, now let's look at the matching condition. So again, we've got our, so this is matching now. We've got our inputs, low and high sensory detail. We've got now a matching prediction. And so if you look at the result, the sharpened representation, what you see is you get a further boost to the representation of clay. So I think this is, I don't think I need to explain this any further. I think this is the more intuitive model. And it just follows from this equation, really. So if you increase the heard speech component, you get a better representation on the outputs. If you also increase the predicted component, you also get a further boost. Okay, so this is just summarizing what the outcome. So matching expectations and increasing sensory detail should have additive effects on neural representations for a sharpened signals model. Okay, so what about prediction errors? So this is the more tricky one. So let's go in the same order as before. Okay, so we've got a mismatching condition, got low and high sensory detail on the inputs. We've got a mismatching prediction, and these are the resulting prediction errors. And so if you can make out that as you go from low to high sensory detail, you have more of this clay. And the reason for this is if you provide more inputs, better speech inputs, but you don't accompany that with a matching prediction, then your predictions remain, or rather the speech input remains unexplained by your predictions, right? So, you've got this clay here, but you're not subtracting anything from it because you have a mismatching prediction. And so this follows then that in the result, there'll also be a clay. So that's why you have this increase in the representation as you go from low to high sensory detail when your expectations are mismatching. For the matching case, this is what you get. So again, low and high sensory detail for the inputs. Now you've got a matching prediction, and these are the resulting prediction errors. And so as you can see here, in the matching condition, when you have high sensory detail, you have very, very small prediction errors. There's hardly anything there. And this is because you now have a clear matching prediction that comes in perfect or almost perfect alignment with the inputs. And so the two cancel each other out. And so you're left with very small prediction errors. And so this is why for the matching condition, as you go from low to high sensory detail, your representations diminish. So hopefully, I wouldn't be surprised at all if this was, this is still unclear. And I think probably it's worth coming back to later in the talk, and I can revisit this and try and unpack it a bit further. But for the moment, I think I'm going to stop there with the explanation and move on to the data. Okay, so yeah, this is just a summary of what I've said before. These are the two models predict in terms of neural representations. But the key thing here now so far is that on the y-axis, we're looking at speech representations, but how exactly are we going to measure those neural representations of speech? So we're going to do so by using MEG, Magneto-encephalography. And so it's very much like EEG, you're just measuring the magnetic counterpart to the electric potentials. And so, for the study, we use this machine here. This is the scanner that was used at the CBU until recently. They've updated they've got a new machine now, but this is the model that was used for this study. And with MEG, you're blessed with a lot of sensors. So for this machine, you have 306 sensors in total, and you've got two types of sensors. So you've got 102 magnetometers, 204 planar gradiometers, and each of these sensor types have complementary strengths and weaknesses, but in general the planar gradiometers have higher SNR, higher signal to noise ratio. So this is what I tend to use, and in the data shown now, it's just data from the planar gradiometers. And I put this sensor space and source space pictures here just to make the point that, you know, when you do MEG analysis, you have this option. What do? You can do your analysis in so-called sensor space. So this is really in the original space in which the data has been collected, right? So it's not in the brain, it's on, you know, on the head. But you can also do source space analysis. So you can transform the sensor space data into the brain, into source space, using mathematical models. So this is nice. You can have a more precise estimate of the cortical generators. But the downside is that if you change the mathematical models that you're using, you change the data, because each model has different assumptions. If you change those assumptions, then you get different data. And so that makes things a bit messy. So, you know, this is one of the reasons why I tend to favor sensor space analyses, and that's what I'm going to be showing you today. Just bear with me a sec. I'm just getting a bit thirsty. I'm talking too much. Okay. Okay, so in terms of the analysis then that we use for this MEG signal, we're not using conventional ERF or ERP method. What we're using is essentially based on linear regression. So this is a powerful method to relate the features of a stimulus to the neural responses. And so in this example here, the features of the stimulus that we're going to be looking at is a spectrogram, right? So I've talked about spectrogram before. So the spectrogram is going to be derived from our audio clips that we've presented to listeners. And this is going to be then, this spectrogram is going to be the predictive variable. And we're going to use that to predict the neural response. So the neural response is the dependent measure in our regression, okay? And it's called time shift regression because we don't know really the exact lag between the stimulus and the neural response. So an easy thing to do then is just to run this analysis for many different lags. That's why it's called time shift regression. And that's why this picture here, it's a spectrogram, but it's multiple copies of the same thing because they're expressed at different lags. Okay, so we got this regression model here, and you know, these are going to be, our betas are going to be there in the model. And what we're going to get out of the model is going to be a measure of prediction accuracy or model accuracy. How well does the model predict the neural response? And so the way we do this is we partition the data into a training set, into a testing set. For the training set, we fit the regression models, so we estimate the betas. And once we've done that, then we use those trained models to come up for predictions for some unseen data in the testing set. And so then what we can do is to correlate the prediction of the model with the observed data in the testing set. And then we can get this measure of model accuracy. So this is now the important bit. This is the outcome of the analysis that we're going to be looking at, because the magnitude of model accuracy captures how strongly a given stimulus representation is reflected in the MEG response. So this is going to be our main dependent measure, the model accuracy. But we didn't just test one set of features of the speech signal, because potentially the MEG might reflect many different speech features. And so what we did was we tested a range of different speech features. So this is depicted here. So in this example, you know, this is one example word in our stimulus set, the word cape. So this is the audio, and all our features are derived from this audio. The first model that we tested was this envelope model. So this is a very simple acoustic model of speech. This is just capturing how much energy there is and how that energy changes over time. So this has been used in the past a lot to some success at predicting neural signals, neural responses. We also tested a spectrogram, which I've talked about already. A spectrogram can be considered essentially the same thing as an envelope. All you're doing differently is you're breaking down the envelope by different acoustic frequencies. Now, both the envelope and spectrograms thought to be a good model of what's going on subcortically or in primary auditory cortex. We've also tested a more complex acoustic feature called spectrotemporal modulations. I'm not going to say too much at this point about this model, but all I'm going to say is that this model, this more complex acoustic model, is thought to be a good account of what's going on in primary auditory cortex or the neighboring auditory areas. And finally, the final thing we tested is not an acoustic model, but a linguistic model, so based on phonetic features. So whether something like voicing is whether the speech sound is a voiced or unvoiced sound. So if it's voiced, the vocal cords are vibrating. If it's unvoiced, the vocal cords aren't vibrating. So it's stuff like that. So it's a purely linguistic categorical model. It's not acoustic. And there is prior work, ECOG work, showing that neurons in the superior temporal gyrus are really well tuned to these phonetic features. So this is one of the motivations for testing this as well. Okay, so those are our different models. First thing then we can do is to look at the model accuracy for each of the different speech features that we have, and to look at how things look at in terms of topography, so over different sensors. And the topographies that you see is very, very typical for an auditory MEG study. So this suggests then that all these speech features correlate with activity in the superior temporal cortex. To say whether it's primary auditory cortex or some other region, in those specific terms, it's a bit more difficult. But we can at least say it's, this is probably reflecting activity in the superior temporal cortex. Okay, so the other thing as well to say about this is that you can see it looks really, really similar for all the different speech features. So they're all occurring in the same sensors. So the more interesting panel really is this panel here. So we're looking at model accuracy and comparing the different speech features, and you can see that really the model that wins is this complex acoustic model, spectrotemporal modulations. So this is the model that does the best job at predicting MEG responses. And so what are spectrotemporal modulations? So to explain this, let's first consider vision, because I think this may be a concept that's more familiar to people. So if we take an image, and in this case of the good old Bridge Cafe, what you can do is to filter the image. So you can just keep the high spatial frequencies or the low spatial frequencies. And we know that visual cortical neurons are attuned to certain spatial frequencies. So filtering the image in this way seems to make sense in capturing how visual cortex represents visual inputs. And so with sound, we can do exactly the same thing. All we're doing is we're going to be replacing the image of the Bridge Cafe with a spectrogram. But otherwise, you do exactly the same signal processing. So you have your spectrogram, you filter to keep just certain spectrotemporal modulations. So in this case, we're using a filter that really likes fine changing spectral information over the y-axis, but it doesn't really care much about temporal information. And so once we do this, then we can summarize things by integrating over time and frequency and putting it here. So we're going to be building up an image that summarizes the spectrotemporal modulation content of speech. And so we can do this using different filters. And so we've got 6 filters here, and that's what these 6 boxes here correspond to. And so just by way of example, if we consider this other filter here, this is in many ways the reverse of the first example. So this is a filter that doesn't really care about spectral information. It's more concerned about temporal information. And so just to give you some sound examples. So let's start with some clear speech.
A good attitude is unbeatable.
Okay, now we're going to be filtering using this filter. So now we're going to be it's going to be tuned to fine changing spectral information, but very slow changing temporal information. So this sounds like this. So I hope you have a sense of what's going on here. I think you can hear that it sounds temporally blurred. And so that's exactly what that filter is doing. This example here, we're doing the reverse. So it's not caring about spectral information. It cares more about fast changing temporal information. So, I think you can hear that it sounds a bit like vocoded speech, because in vocoded speech, just like with this filter, it's reducing the spectral detail. And what that does is it makes it sound like a harsh whisper, because many important details of the voice, such as pitch, is conveyed by spectral information. So if you reduce that, if you destroy that spectral information, then you destroy those typical voice qualities. Okay, so now that we've got a handle on what the MEG signal is reflecting, so it seems to be to do with the spectrotemporal modulation content in speech. So when I talk about neural representations of speech, I can really be more specific. It's neural representations of spectrotemporal acoustic structure. So now that we know what the y-axis is now, we can return to this question of sharpened signals versus prediction errors. And we can test now, we can see how the model accuracy measure in our regression model for the spectrotemporal modulation model, how that differs between conditions. And so this is really the key figure of the talk. So what you're seeing here, I'm splitting things by hemisphere, so left hemisphere, right hemisphere. And so we're looking at this model accuracy measure as a function of our conditions. And I think you can see that in the right hemisphere, there's not much going on, but in the left hemisphere, as you increase the amount of sensory detail, you get enhanced neural representations of spectrotemporal structure. But then when your prior expectations are matching, you have the reverse. And so if you remember, that's really very much like that prediction error model. So this result really is consistent with the prediction error model, and it's inconsistent with the sharpened signal model. Okay, so we can go a little bit further than this. So with this time shift regression technique. So far, what I've been describing to you is a so-called encoding model. So it's called an encoding model because our stimulus is assigned to be the predictive variable, and the dependent variable is the neural response. But we can flip things around so that our neural response becomes the predictive variable, and the stimulus is now the dependent variable. And it's called a decoding model, or also called a backward model, because really we're going backwards in time. We're starting off with the neural response, and then we're going earlier in time backwards to try and decode the auditory stimulus. And so, we're going to do this not just for the fun of it. There's a reason for doing this, and one of the good things you can do with a decoding analysis is that instead of looking at model accuracy broadly, we can really break it down and link model accuracy to specific spectrotemporal modulations. And so I'm going to spend some time talking about that. But first, I think we need to return to the acoustic signal. Before I present to you the MEG data, I need to go back to the acoustic signal because I haven't actually shown you what vocoding does to spectrotemporal modulations. All I've shown you is the spectrograms. So let's look at the images of spectrotemporal modulation content. So this is what vocoding does to spectrotemporal modulation. So here, for comparison, we have clear speech, and here we have different vocoded versions of the same speech inputs, going from 24 channels all the way to 1 channel. And to visualize what's going on here, it's helpful just to do a direct comparison, so a subtraction. So let's take 24 channels and subtract from that 1 channels. And so here, what you're seeing in red is that when you have increased sensory detail, you have higher magnitude spectrotemporal modulations in this 2 to 8 hertz range. So temporal modulations that lie between 2 and 8 hertz. Okay, so this is helpful, and it makes sense actually because lots of prior work has shown that these temporal modulations are very important for speech intelligibility. So this explains why vocoding has such a dramatic impact on speech intelligibility. Because if you remove or affect the signal in these temporal modulations, then you're going to have consequences for speech intelligibility. So that's purely on the acoustic side. Now we can compare this to what's going on neurally. Okay, so I'm just going to bring back the acoustic signal just for comparison. That's what I've just shown you. Now we can look at the MEG. So what I'm doing here is now I'm plotting model accuracy, but now with decoding we can look at each spectrotemporal modulation separately. And just to help with the visualization, I'm going to be doing computing simple effects to capture the interaction. So I'm going to look at this comparison of 12 minus 3 channels for the mismatch case. And as I've shown you before, as you increase the amount of sensory detail, you have an increase in model accuracy, but then it turns to blue, so it goes negative for when your prior knowledge is matching. Okay, so this is just the interaction, but the kind of informative thing about this analysis is looking at where stuff is happening in terms of spectrotemporal modulations. And as you can see, these are manipulations. Experimental manipulations are not affecting all spectrotemporal modulations equally. It has a really targeted effect, and it seems to be most pronounced for 2 to 4 hertz, which overlaps to some degree to what we see in the acoustic signal. Okay, so all this is saying, and it's very helpful to show this, that our kind of effect of prior expectation isn't just a random influence on spectrotemporal encoding. It's really tightly linked to the degradation that vocoding does to the speech signal. Okay, so final thing. Other nice thing you can do with decoding is to look at the time course of when things are happening. So instead of aggregating over different lags, we can do a single lag analysis and get a model accuracy for each lag separately. And the critical line to look at is the black line. So this is capturing the interaction effect on model accuracies due to sensory detail and prior knowledge. And as you can see, certainly within 100 milliseconds, we see an effect. And it may be even as early as 25 milliseconds. So that's when things are significant for the interaction. So this, again, this is not only are we finding evidence for prediction errors in terms of that question of when prior expectations are combined with speech input. Well, this analysis suggests that really prior expectations are combined at early sensory stages. All right, okay, so it's a lot to get through. I hope you're still with me. This is out there already as a preprint, so this is the link to it if you want to learn more about that study. It's currently under review. Hopefully it will make its way to a journal soon. So I'm going to leave that link up, and I just want to summarize everything I've just said. So I've shown you that prior expectations and sensory detail have interactive effects on neural representations, which is very consistent with this idea of prediction errors. And these interactive effects are tightly linked to spectrotemporal modulations that are affected by the speech vocoding procedure. And it also occurs within 100 milliseconds of speech input. So this is consistent with this idea of prior expectations influencing early stages of processing. Okay, so I think I'm probably around 40 minutes now. And I'm going to be just telling you a little bit about some more recent work. And it's really to give you a brief idea of where things are headed. So I've come to Sussex, and one of the things I want to do is to build on these ideas, testing prediction errors from other models, but going towards more naturalistic prediction. Because this paradigm of using written text, it's been really helpful for us. We've used this paradigm in lots of different studies. But it's really not really reflective of everyday listening unless you find yourself watching TV with subtitles on. And so what I want to do is look towards more naturalistic prediction. And so one of the things, one of the places you can look for in terms of naturalistic prediction is speech itself. You don't need to turn to written text. You can look at speech itself because some words are spoken more frequently than others. And so that might convey stronger predictions or weaker predictions, depending on the word that you're hearing. Okay, so in this study, we exploit this. So this is actually an fMRI study. I'm going to be telling you very briefly about this. So yeah, in the study, we're using word frequency to derive measures of how strongly speech is predicted. Okay, so we have this stimulus set consisting of these bisyllabic items like quota, delta, cactus, status, etc. And so what we've done is to compute conditional probabilities. So what's the probability of hearing the second syllable given the first syllable? And this is all based on word frequency. So we've got these word frequency measures from a linguistic database. And so this has allowed us to calculate the predictability of these second syllables. And we've constructed our stimulus set. So half the items have a second syllable that's very strongly predicted by the first syllable. The other half of the items, the second syllable is only weakly predicted by the first syllable. And then analogous to the mismatch for the written text paradigm, we also have match-mismatch here. The way we've done this is to cross-splice our items. So for the mismatch items, they become pseudo words. And so, for instance, if you hear kak and you're expecting tus, but actually you hear ta in the end, it's a mismatching prediction. This allows us then to factorially manipulate two forms of prediction, whether it's strong or weak, and whether it's matching or mismatching. And so, as I said, this is an fMRI study. And so for this fMRI study, it made more sense to use a technique called representational similarity analysis. And so with this technique, then we can look at multivoxel patterns, and we can look throughout the brain and we can see, we can ask, okay, so for a pair of items, where are there multivoxel patterns that are more dissimilar when the second syllable is different compared with when the second syllable is the same in the pair of items, right? And so if you do this sort of analysis, then we can look over the brain, and it turns out that the primary auditory cortex, Heschel's gyrus, robustly represents syllable 2. And so once we've done this, then we can do a region of interest analysis to look at things by condition. And what we see is a robust interaction between strong versus weak prediction and matching versus mismatching prediction. And so all I'll say about this is that, again, this is really speaking to this idea of, you know, when prior expectations combine with the speech input. So this study with fMRI with more precise spatial resolution will suggest that it really is very early on. Prior expectations can really modulate early sensory stages of processing, even in primary areas. And so that study has been done. Again, that was done at the CBU. So for Sussex, then, I'm going to be pushing this naturalistic prediction idea further, because there's really nice work, recent work that have used the linear regression technique I was talking to you about for the first study, and they're applying it to audio books. So listeners in these experiments hear these audio books, so very naturalistic. For some reason, it tends to be Charles Dickens all the time, but I think it doesn't need to be. But they hear these audio books, and they do this linear regression, so they're correlating different speech features with a neural response. And in this study here, this is Broadbeck et al. In current biology, they're using actually a measure of prediction error in the speech signal, relating that to the neural response. And then there's also this measure from information theory called entropy. It's another form of predictability. And they see neural responses encode these variables, but you know, I think there's a lot to be done here, because one of the things that hasn't been done in this work is that they've never actually tested whether prediction errors or sharpened signals better explains the neural responses. Okay, so probably went too quickly, but I've finished. All I want to say is that just to say thanks to Matt Davis, who was my postdoc supervisor at the MRC CBU, and also the MRC for funding this research. So yeah, that's me done. Thank you very much.
Thank you very much, Ed. That was a fascinating talk. We have some time for some discussions. There's a couple of questions in the chat, so I'll try and read the questions out, and if I butcher the questions, please jump in. So Maxine is asking about the filtering that you did, that was done, and was that done to the actual stimulus that you presented to the subjects, or was it done to the model betas?
The filtering.
Yeah,
so the filtering was done. Okay, so let's turn to this slide here. Yeah, okay, so in terms of filtering, so we haven't actually. You can do that. You can. So that's what's. Yeah, so that's. I can see the confusion. So this is what's done in this study, and actually this is what I want to do: is some experiments to look at perceptual learning. You know, how well do you get? You know, how people get better at understanding speech over time. And I think this tool here, spectrotemporal modulations and filtering them, is a nice tool to explore that question. But we haven't done that exactly. So what we've done is use vocoding. So we've done noise vocoding, which is not really directly filtering in terms of spectrotemporal modulations. So there's no Gabor filters that are tuned to specific spectrotemporal modulations. But the result of what you get with vocoding is very much like the results you would get if you tuned with a filter that was tuned to more coarse spectral information. And so that's what we did. We used vocoding, but it doesn't matter. We can still visualize what things look like in the spectrotemporal modulation space. And then this sets up the analysis where we compare this to the neural data.
I see. Thank you. That makes lots of sense. Could I ask one more question?
Sure.
Because it was really cool and you have lots of interesting analyses. When you do the one with the lag, so you're presenting model accuracy over lag.
Yeah.
So there's an analysis called temporal generalization. I don't know if you know where you're kind of.
Yes.
Yeah. Is that basically the same as this? Or
No, I don't think it is. No. So So yeah, with temporal generalization analysis, which tends to be done with, the RSA approach, that's asking the question of whether spatial patterns at one time point are the same. Is it the same pattern at another time point? I mean, I think that's what it's doing, and that's this analysis is not doing that. So what it's doing instead, I think that, to put it in words, it's more about
So is it that what you're decoding is different? You're not necessarily decoding the pattern of activity, but you're still looking at where in time that information is.
It's where in time. Okay, I found a way to put this. So with this analysis, okay, so here, if you can see where I'm pointing. So right, this is saying that using the neural responses that we're seeing, we can decode spectrotemporal modulations in the speech signal. And at this time point here, we can also do that. It's above the threshold, so that's also significant. But it doesn't mean that what's going on here is the same as what's going on here. It could be that different features of the neural response, and maybe even different acoustic features, are what's driving this data point here compared with this data point.
Yeah.
So that, in that sense, that's different to the temporal generalization analysis.
Yeah, I understand. Yeah. Thank you.
Okay, and we've got Sam asks, to generate the betas for the decoding model, do you just invert the betas of the encoding model, or do you retrain? Retrain.
So they're entirely separate models. And
thank you very much.
Yeah, no worries.
Okay, and Tom asks, I like your conclusion about prediction errors because there is a hint from postsynaptic protein computation that individual synapses can act like temporal recognizers or predictors. I guess that's just a comment.
Sure. I mean, I'd be happy to be pointed to that work. I'll get in touch. So, sorry, who?
It's in the chat. It's Tom Cabaz.
Okay, great. Yeah. Okay.
Hi. Yes. Sorry. Drop me an email
yeah, sure, that'd be great. Thank you.
I put my email address in the chat now, and then drop me an email and we can discuss.
That sounds interesting. Yeah, great. Thanks a lot.
Thank you.
And Jamie has a question. You mentioned 4 models of speech representation and said that one was best, but perhaps the best model changes over time.
Yes. No, that's true. That is true indeed. That's potentially true. Yeah. Yeah. So I mean, with this analysis, you're constructing the predictive variables where you're using multiple copies of the stimulus at different lags, and then what it's doing is integrating over those predictors, right? So for each neural response that you're seeing at time point X, it's going to be the model's going to kind of come up with a prediction for that data point based on the speech signal in the past over those lags, and so it's integrating over all lags, and so it's kind of a very it's a broad summary measure, and it may be that if you break it down by different lag windows, then yeah, different models might you might get a different outcome. That's true. Yeah.
And Dan Campbell-Michael John asks, wonderful work, Ed. It would be fascinating to see how these naturalistic effects change at different stages of life, particularly in the very young and the very old. Would you expect things to change?
Oh, that's an interesting question. Yeah. So, yeah, no, individual differences is something also I want to explore. I mean, I remember Peggy series, I think, came to give a talk, Sussex, of the Leverhulme series of talks, and she was presenting work where she was asking this question of there's a hypothesis that in autistic individuals that there's a difference in how prior expectations are combined with sensory inputs, but it's not clear whether it's to do with overly precise sensory inputs or weak priors. I think it's that way around. And so, yeah, she was presenting work that spoke to that question. And I think you can ask a similar question to younger versus older adults, or even, within younger adults, there's a lot of variability in how well people do at understanding speech in difficult listening conditions. And so why is that? And so it may be that they're less reliant on prior knowledge, or over-reliant, or something to do with sensory input. So if you can, that seems like a very interesting question to ask, to try and ask what's going on. Is it to do with the expectations and differences in how expectations are exploited during perception, or is it more the sensory input side? And I don't know really off the top of my head with the younger versus older adult question, but I think, yeah, that's very interesting.
Okay, and Corn asks
Cornet.
I do apologize.
No worries.
Any ideas from your work for improving processing in cochlear implants? By the way, everyone, feel free to unmute your mics and join in the conversation if you desire.
Yeah. So no, that is. I think in the short term, I'm going to be. I think the experiments will be done with normal hearing listeners, but I think that's always a question for the long term. It's just, I'm afraid, long term at the moment. But yeah, I think that you'll know better than I do. But there's a lot of the variability that you see in cochlear implant outcomes. A lot of it is biological, it's technological. But from what I can see, that even if you take into account those factors, there's still some variability left unexplained. And so, there's a hypothesis that a lot of it's to do with cortical plasticity. I've done some other work with perceptual learning, where the perceptual learning is really tightly linked to your expectations, how you use your expectations. And so, if we compare a listener who doesn't do very well with a listener who does really well with their implants, perhaps asking that question again. It relates to this younger versus adults question earlier as well. Perhaps by considering this framework, we can get a better idea of what explains individual differences in outcomes. So it's some involvement for cortical plasticity, but is it can we say something a little bit more specifically about whether it's how listeners are using expectations to update their internal models?
Yeah, I think the models that feed into cochlear implant design are probably fairly naive at this stage yet. And I'm impressed that you built up a more detailed picture that can tell you more about what's really going on in auditory cortex, for example.
Yeah, sure.
Great.
Thanks.
Okay, we'll make this the last question from Sophie. A rather general question, but I always wonder to what extent prior expectations overlap with top-down attentional settings. For example, in the green needle case, I think that once expect that both sounds are present, you can selectively attend to each one with the same result.
Yes, no, very, very astute question, Sophie. And this is one of the questions in the field. So this predictive coding literature. I guess, one hypothesis that is if it's strictly about prediction, if it's strictly about statistics in the input, so about Bayesian inference, then really this influence of prior expectations should be independent of attention. So all the effects that we're seeing, according to that hypothesis, it shouldn't matter whether you're attending or not. I don't think that's true. I think, there's evidence to suggest that, this, whatever measure you're considering, whether it's behavioral measures or neural measures, they're really very much modulated by attention. And there are experiments that try to separate out the two. So they manipulate factorially attention, and they do the same with prediction. And so they try and kind of separate out those contributions. And well, the work I've seen is that there is always an interaction between the two factors. So I think it is tightly linked with attention. And in fact, this last work I showed you, I think I've mentioned this to you, Sophie, before, that, so you've got this naturalistic speech, and it seems like the predictability of the speech input is correlated with neural responses. And if you actually adapt the paradigm, so instead of presenting one speaker, you present two speakers. So it's now a selective attention sort of paradigm, and you're told to, the listener's told to just focus on one speaker, so the male speaker, and or let's just say the female speaker, and ignore the male speaker. And then what they show with that sort of paradigm is that this effect that they were seeing previously for phoneme predictability or prediction errors for the single speaker case is completely abolished when you're not attending. So, and again, I think there's more work to be done, but I do think that, I mean, the short answer to your question is that I think you can define prediction and attention to be different, but and you can construct experiments where they're manipulated separately, but that's not to say that they don't interact together.
You're muted, Sophie. I can see you.
I just was saying, okay, thank you. Yeah, thanks very much. That's really interesting.
Thank you very much, Ed, for an absolutely fascinating talk, and thank you everyone for coming along. This is the first of a series running weekly on Tuesdays at 1 p.m. And next week we have two new fMRI project proposals from Julia Kubai and Jamie Ward. So hope to see you next week. And thank you, everyone. And thank you again, Ed.
Thanks. Cheers.